Now in the last video I talked about the structure of the mitochondria, and now I'm going to cover the functions of it. So first of all, let's talk about the function of the outer and inner membrane of the mitochondria. Well, the first one is protein transport, right? So transporting proteins through these membranes. And we need these proteins to get kind of transported, and you'll see why later in my video. But just understand that we need to move proteins inside the mitochondria, eventually inside the matrix. And so let's talk about how that happens. Well, first of all, as I said, inside the matrix we have mitochondrial DNA. But mitochondrial DNA is only responsible for 15% of the proteins. So you need actually 85% of the proteins from the nucleus. And so from the nucleus we'll have DNA, right? Then mRNA, then leaving the nuclear pores, then attaching to the RER or ribosomes freely in the cytoplasm. Protein synthesis will happen, and boom, you will have these proteins moving to the mitochondria. Okay, so mitochondria has special transporters which can move these unfolded proteins through the mitochondria. This is what you see here in the left picture, and it will move these unfolded proteins through a specific receptor, this outer membrane translocase receptor called TOM, also called TOM receptor, and move the protein into the intermembrane space. Now in the intermembrane space we have chaperone proteins. These chaperone proteins are responsible for moving it once again to or through the inner membrane to the matrix. And so this new channel opens, this tip channel opens, and it'll press this unfolded protein inside the matrix, where it can then be utilized as an enzyme in metabolic processes, right? And from here we have different enzymes, proteinases, which will then again fold the protein in a kind of specific way, which will activate the protein, and it can be utilized, for example, in metabolic processes. Okay, so this is the outer and inner mitochondrial function of protein transport. You're moving proteins through, right, to the matrix eventually. Now let's talk about mitochondrial electron transport chain, which is actually what makes it the powerhouse of the cell. So the electron transport chain is primarily found in the inner membrane of the mitochondria. And here we have protein molecules kind of make or a part, these protein molecules are part of bigger protein complexes. This is what you see here, protein complex 1, 2, 3, 4, so on and so forth. And also attached to that we have ATP synthase. And so what will happen is, remember that in the Krebs cycles, the final products are FADH2 and NADH2, right? FADH2 and NADH. And these carry loads of electrons, and these will take electrons and move them to the protein complexes. And they will transport it to these protein complexes, will move them from the complexes to from a chain of high energy to low energy. And every time they do this, they will pump protons into the intermembrane space, and the protons will then move through ATP synthase, where it will make energy by fusionating ADP, adenosine diphosphate, and phosphate, making ATP. And then we have made ATP. So you're generating ATP through the electron transport chain, otherwise known as the oxidative phosphorylation. Okay, right? Everything to do with the Krebs cycle, NADH, FADH2, carrying electrons to the protein complexes, then hydrons to the intermembrane space, moving through ATP synthase and fusionating ADP and phosphate to making ATP. Right, and this plays a key role, as you can imagine, in cellular respiration. Now we go on to mitochondrial function of the matrix and mitochondrial DNA and ribosomes, right? All these metabolic reactions, and I'm going to talk about some of the most important ones, right? The first one is with glucose. So glucose is taken up by the cell through glucose transporters in the cell membrane, and inside the cell, glucose is then converted to glucose 6-phosphate, G6 phosphate, or G6P, by the enzyme hexokinases. So glucose in the enzyme will be converted to this, and then this is then metabolized through glycolysis to pyruvate in the cytoplasm. And then you'll see that pyruvate, very, very important, guys, will kind of get moving into the cell. It will get moving into the mitochondria. The pyruvate will move into the mitochondria and get converted. Once again, pyruvate will once again get converted, right? Before we said glucose to pyruvate, this is called glycolysis. Now we have pyruvate going inside the mitochondria, and now it becomes acetyl-CoA. Acetyl-CoA is of course a part of the Krebs cycle, and it will undergo changes through the Krebs cycle. Eventually you'll make these high energy molecules that are carriers for electrons, NADH and FADH2, which will be made, and then that will go to the electron transport chain to make ATP, right? So pyruvate to acetyl-CoA to Krebs cycle to eventually making NADH and FADH to the electron transport chain, and then making ATP. Now another reaction is fatty acids, which can be brought into the matrix of the mitochondria, can go through metabolic reactions and become acetyl-CoA once again. And this is called beta oxidation. So beta oxidation is with fatty acids brought into the matrix of the mitochondria to become acetyl-CoA. This is called beta oxidation. Then we have a bunch of other things. We have amino acids which can be brought into the mitochondria, where they can get metabolized as ammonia, where it can give its product away in the Krebs cycle. Then ammonia will go through a series of reactions and releasing a product called urea. Now urea is less toxic than ammonia, and this is easier for it to get excreted. This is called the urea cycle. The urea cycle is also a metabolic process inside the mitochondria. Then we also have glyconeogenesis. We have ketogenesis, which is also a product. I'm not going to talk about these ones too much in detail. Apoptosis is programmed cell death. I want you to understand that inside the mitochondria we have cytochrome C inside the mitochondrial matrix, and we don't want this cytochrome C to leave the mitochondria, right? And this is controlled by a protein called the BCL2 protein. Now, of course, if the BCL2 proteins are reduced, then cytochrome C molecules will come outside the matrix and of the mitochondria and inside the cytoplasm of the cell, right? This is very bad because it will activate enzymes called capsases, which are proteases, which then will rip in different cells and organelles, and therefore you get apoptosis. Okay, so mitochondria is actually responsible for apoptosis by BCL2 being reduced, and therefore cytochrome C leaving, and now you'll have apoptosis. Now lastly, let's talk about the endosymbiotic theory, which is a widely accepted scientific theory that explains the origin of eukaryotic cells, which are cells that contain a nucleus and membrane-bound organelles